All right. Do you all have the Firefox plugin and this mirdb.sqlite? Yeah. Okay. Um, so I need to have it. So from your Firefox, you can open the SQLite database that you downloaded. Wherever you downloaded it, you have to browse into that folder and open it up. So we have one table called mir to target. This other table, SQLite underscore sequence, it's an internal table for SQLite system. You can ignore it. Okay, so you don't have to really work with it directly. All right, mir to target is empty on my computer. And I need to wait a long time before it downloads. Did you have to wait that long too? Yeah? No? Half an hour. That's longer than I want to wait. I just need to go to a computer that has the database already. Okay. MIRDB, okay, good. Okay, so we have this first table, the only table in this database.
How do we load the table?
So how do you load the table? If you go to, how do you open the database? If you go to database and then connect database, or if you click on this icon that has the open folder picture. Yep.
And we use
mirdb.sqlite. Yep. All right. So we have a single table that has the ID field that's numerical and it's auto incremented. We have the microRNA name. So microRNAs are short RNA molecules about 25 nucleotide long, and they go and bind to messenger RNAs and repress them. So they repress the translation of messenger RNA into protein. And effectively, they regulate the amount of protein that the gene would produce. For especially in plants, after they bind, they cause degradation of the mRNA. But in animals, it usually suppresses translation, meaning when you have the microRNA, too much of one microRNA, it goes and binds to its targets, and you get less of the protein products from those genes. This binding is based on complementarity, just like the base complementarity at the DNA level. So A complements with T, G complements with C. You have the same complementarity at the RNA level, even though it's not as you don't form double helix, but you still have complementarity that they form a strong bond. So microRNA, whenever the sequence of the microRNA is complementary to the sequence of a mRNA, then it binds to it, and then that binding causes suppression of the translation. Essentially, that mRNA cannot really be processed in the ribosome to produce protein. Okay? So microRNAs are regulatory. They are a regulatory mechanism that control how much of each gene is being translated into protein. Transcription factors is another mechanism. So when you have a transcription factor, they can activate or repress the transcription of the gene, whereas microRNAs act at the translation stage. After the mRNA has been produced, they suppress its translation into a protein. Okay? So there are computational methods that predict which genes a microRNA would target. The complementarity doesn't have to be 100%. So some of the nucleotides in that 25 nucleotide sequence may not match. But even with some mismatches, you may still get a strong binding and have a suppression effect. And some of these microRNAs are conserved in the evolution, meaning we have them. Mice would have the same sequence of microRNA and so on. So using the sequence information and the conservation information, there are computational methods that predict which genes a certain microRNA will target. Okay. So one of these methods is called MIRDB. And after they do their predictions, they deposit these predictions, their results into a database, but they give you this database in the form of a text file. Okay. So I already downloaded the text file. I read each line of that text file and then converted it into a row in this table. So I did all the construction of the table and insertion into this table, which you will do after we go over the select statements. All right, so we have the microRNA field that lists the names of the microRNAs, and you can see that it repeats because one microRNA can target more than one gene. We have the gene ref-seq ID field. Sometimes you may have the gene symbol, but MIRDB text file did not provide the gene symbol. They provided us the ref-seq, which is the reference sequence identifier. Okay, so we have a number of different identifiers for genes. We have the gene symbol, ref-seq ID, Entrez ID, and sometimes when people build a new database, they may have their own ID for the same gene. Okay, and there are some reasons behind why they do it. And we also have a score where the higher the score, the more confident that MIRDB is for this binding. So a target score of 100 would mean that they are pretty certain that the microRNA is targeting the gene. And they don't list all their predictions. I think the cutoff is 50. They don't show you anything that has less than 50 prediction score. Okay. All right, so let's now get back to our PowerPoint. And here's what we're going to do. Have any of you already solved this? How many microRNAs are predicted to target XM539064? Any answers? No. All right, so we're going to write a SQL statement to find the answer. No, all right. And I will give you hints and have you work on it. And the first person to do it will get a bonus point, which will then turn into a grade at the end. So here's how you're going to enter your answer. Once you find the answer, you're going to go to Scratchpad, and let's say I have the answer. Write your name, and then write your SQL statement. If somebody has already started writing, you need to write after them. Okay? So Jennifer would have her answer here. And then we'll go through these answers and see which one is correct. And the first person with the correct answer will get the bonus point. So write your name before you enter your answer. And if somebody has already started a line, don't start a new line above them. So if you know SQL, you may just write it on the Scratchpad. If you don't, it would help if you try it in your Firefox plugin first. All right, so you have a single table, right? That means you need to, you don't need any joins, because joins work when you have, actually you can join with the table itself, but for this problem, you don't need any joins. You need to write a select statement that contains a where clause. And in the where clause, you need to specify your condition. In this case, the condition is that you want the gene name to be XM539064. And we are not really interested in the microRNA names themselves, but we are more interested in their count. Okay? All right. And we looks like already have an answer. So let's do it here first. So we're going to do a select statement, right? And then let's leave what we're going to get out to the end. We're going to get it from mir to target table. Mir to target. All right. And then we're going to have a where clause where we specify the condition on which we want to filter, with which we want to filter. So where, you could say mir to target dot gene refseq ID, too long. So instead of that, let me go back. Since I have just one table, I don't need to really type its name. When you have more than one table, it's good to write the table name and then say dot the field name. So in this case, we'll just say gene refseq ID equals, what was it, XM in quotes XM underscore 539064, and then close the quote. All right. So you're selecting from this table where that condition is satisfied. You are getting all the rows that have gene refseq ID equals this value. So what are we getting out of this? We need to get not the rows themselves, but their counts. So we are interested in how many. So we need to say count parenthesis. So the count function inside the parenthesis, you could specify any of the field names. So you could say count of microRNA values, or the count of IDs. But it's more efficient to say count star. Okay. All right. So the first answer is correct. We say select count from MIR to target table where gene ref-seq ID is equal to that value. Okay. I'm going to delete the rest, which appear to be correct also. So for the next problem, Apurva cannot type her answer. Okay. All right. So for the next problem, we have how many predicted targets of CFA-LED7A have a prediction score of at least 80. So it's very similar to the first problem, but now you need to have two conditions that you need to AND together. Okay. So once you are first for a problem, you can work on the problem, but let somebody else write their answer. All right, I'm going to have to change this. But let's wait until this is done. All right, so Shweta's answer looks correct. We are again selecting the count of rows from the same table where the microRNA is CFA-LED7A, okay? And the score is at least 80, meaning greater than or equal to 80, okay? So this will get the number of targets of MIR to target for this particular microRNA, and then with a score of at least 80, okay? So that's the correct answer. I'm gonna again delete the rest. Now when you type your answer, don't type it on the scratchpad, copy and paste it from your answer, all right? So only start typing when you have the answer in place on your own computer. All right. So how many printed targets? Let's do that. Okay, so the next one is where you list the microRNAs and the number of their targets. So I'm not just interested in a particular microRNA, but I want to list them all. All right? And when I list them all, for each of them, I want to get their targets, and I actually want to get the count of their targets. Okay? So this needs to contain a group by clause, which I haven't really shown you yet, but you can search Google for it. Okay? And I'll let you use your Google skills to see how you can use group by, and then tell you the answer. Did your computer die? If you sit here, I can plug in this wall. Mhm. All right, as you are working on it, let me give you the incomplete answer. So let's select microRNA and then count of what was it, gene ref-seq ID. You can also say a star instead of gene ref-seq ID. So we are selecting the microRNA and the count of its targets from mirror to target table, and that's it. Okay. If I run this, it will take a lot of. Yep. So it actually ran pretty fast. I only get one single microRNA. So a count function, just like a sum function, is an aggregator, meaning it takes a set of values and it reduces them to a single value. Right? When we summed up the GPAs, it'll give you a single row. It will not give you the individual GPA values. Right? So you're taking all the GPAs, all the rows, and reducing it to a single number, to a single value. Should I try to be louder than her? So what we need to do is we need to group things and then aggregate within the groups. So we need to group all the microRNA. Let's see, the first one is CFA-led7A. We need to group all the rows that have microRNA CFA-led7A, and then apply our aggregator function, which is the count, for those rows. Okay, and then we need to take the second group, which will be, what's the next microRNA? Next. 7B. Okay, CFA-led7B, and then aggregate on that. Okay. So we need a mechanism by which we can take all these rows and group them by microRNA. And that's where the group by phrase comes in. Okay? So if anyone has the answer, yep. So we get one answer. We are selecting the microRNA and the count of the rows in the result from that table, and then we are grouping by the microRNA value. Okay? So it'll take the table, the entire table, group by microRNA means you're gonna take the unique values of microRNAs, and all the rows with the first value, with the first unique value, will be considered as the first group, and any function in your select statement will apply to that first group. Okay, so let's do that. Oh. Add that to the end of our SQL statement, and then run SQL, and it takes a while to run, but we should get for each microRNA the number of their targets. And for the sake of speed, I'll just say limit 0 10. That way I will only show the first 10 rows of the result. Not the first, I'm not processing just the first 10 rows of the table, but whatever result I'm getting without the limit, I will only get the first 10 rows of the result when I apply a limit. Okay? And the limit makes it work best because it doesn't, it's not trying to show you all the results. All right. What if I remove the count function, what do you think I would get?
Oh, you get the pairs of mirror
Will I get the entire table?
gene reps. Well, no, you think you get unique since you're grouping by.
So grouping by microRNA means your result will have unique microRNAs. You will have one row for each different microRNA. But the gene ref-seq ID is sort of, in a way, undetermined, and I believe different database systems may return different results for this. But you will get just one result for each group. In this case, I think it's going to be the last gene ref-seq ID for that microRNA. So it'll group by CFA let 7A. We go to the beginning. And then it will group them all and return and calculate this the value for gene ref-seq ID. And as it's going through all those rows that has CFA let 7A as the microRNA, it's going to keep updating what it thinks the result should be. And then the last one will stay as the result. So of the microRNAs in the table that has the value CFA let 7A, the last one was had the gene name XM5385253. Okay. All right. Any questions? Okay. So for this problem, let me just talk about it now. So in case we don't get to it, let me open it in here. I actually have introduced another table, but what you will get is a table or a text file that links a microRNA to its family, and it'll have many fields, but you don't really need to store all those fields in your database. In my case, I kept the microRNA family name, the species ID, and the microRNA name. So HSA let 7A, 7B, 7C, all the way to 7F, they are in the same family. Okay, the family name is let 7, 98, and so on. Okay, and this target prediction method, when it reports its results, it doesn't report it on a single microRNA level, but it reports it at the microRNA family level. So it will not, it's not going to tell you the targets for HSA let 7A, but it will tell you the targets for let 7, 98, 4458, 4500. Okay. So the other table that you will get from the target scan is mir family to target. I named these tables myself. They don't tell you that the table name should be that way, and you can have any table name that you'd like. And their field names may be different. Again, you can give any field name you would like. So in this one, this is similar to the mir DB table that we looked at, but instead of microRNA, now we have mir family, the family name of the microRNA. And when storing the data in my database, I kept the species ID, the gene symbol, the gene ref-seq ID, and the score. Okay. All right. So what we need to do to find the targets of a particular microRNA is the following. We first have to start with the mir to mir family table. And then let's say we are trying to find all the targets of HSA let 7A. And our code should work for any microRNA, not just let 7A. So we'll search in this table and condition it on microRNA equals HSA let 7A. and then we're going to say and. We're going to take the mir family field and use that to link into mir family to target table. So we're going to say mir to mir family, which is the stable name, dot mir family field equals mir family to target dot mir family. Okay, so we are starting with the microRNA we are searching, and then we are linking those two tables based on the microRNA family. And doing that will get us the targets of this microRNA by linking through the microRNA family. Okay, makes sense? All right. And the mir to mir family will have, sorry, mir family to target will give us the targets of that microRNA. So similar to the joining that we did for students and courses. Okay, so all right. So to do that, we need to construct the database, or you're going to do it as your homework assignment. And I will, for the remaining of this lecture, I will construct the MirDB database that I provided to you, and you downloaded it. Okay. So how do you construct this one? You need two tables that come from two different files. If you go to the link provided here, you're going to download the mir family, which contains the information about which microRNA is part of which family. So that's one of your tables. And the other file is going to contain the target predictions that give you which microRNA families target which genes. Okay. All right. Before I get into constructing the database, let's do this. Let's talk about this Go database, where the database is already there, so it's easier to show you examples. All right, so far you've seen databases that have a single table or two tables. Let's look at a more complicated database. Yes. All right, so this is a graphical view of a database. Okay, so each big box represents a table, and at the top you have the table title, the table name, and within that box you have the list of fields in that table, the field names. Okay, this entire graph is called an entity relationship diagram. Each table can be referred to as an entity, and the links, these lines that connect two tables, are called relationships. So we may have one-to-many relationship, many-to-many relationship, or one-to-one relationships. Okay. All right. So it's a big database, but we will only focus on as I think 3 tables here. So gene product table gives us information about genes, the gene name, the gene ID, and then we have the association table, which will link one term with another one. Okay, let me actually go over the example that I'm asking you, so I don't confuse you with all the other things on this graph. Here it is. Okay, so we're going to start with a simple example that does not involve any joins. We're going to retrieve the names of the species that are under the genus, I don't know how to pronounce that one, Drosophila. So we are going to get all the Drosophila species. So let's do that. We're going to go to this third link where we can enter SQL statements. Okay, we're going to select from the species table. Which one was the species table? Yep. Species, and then say where that equals Drosophila. And then go down, hit query. It'll take a little time. I have another outlet available if you guys want to sit at the front, if any of you is running out of battery. Right here, yeah.
It's the outlets in the tables don't seem to work.
Yeah, we figured that, yeah. I'll have to complain to them. I was very excited that they had outlets here.
Thank you.
Sure. All right, and I think we got our results. If you go down, so this gets you all the fields. I didn't do any field selection, but we get the Drosophila and the species. One of those will be the fruit fly, which is the melanogaster. Okay, so we have a lot of them. So since we only want the names, let's get species dot species. So the name of the table is the same as the name of the field, which could happen. Okay, and since you have a single table, you don't really need to provide the table name dot part. You can just say select species. When you have more than one table and they contain the same field names, you have to specify which table a field is coming from. So now I did the same query, but instead of getting all the field names, all the field values, I'm just getting the species column. Okay. All right. So that's the first one. Next one, we want to retrieve the genus and the species name of the organisms whose species name has a prefix MEL. Okay. So when we say something equals a string, we are comparing the strings in their entirety. Like here where we did genus equals Drosophila, we want the entire value of that field to be equal to Drosophila. If we say species equals MEL, we're not going to get any results because there's no such species. The question asks us to get all the species that whose names start with MEL. Okay, so we need to rely on a function in SQL. Most functions come in parenthesized form, but this one comes with a phrase that says like. So instead of the equal sign, we're gonna say like. So where species is like MEL and then percent symbol. Percent symbol means match anything after MEL. You can also match the beginning of the string where you could do percent MEL. Okay, but for this problem, we want the species to start with the letters MEL followed by anything. And let's get species dot. Let's not that. Oh. Let's run the query. And we get all those species whose names start with the letters MEL. Okay. All right. So next one is going to be more complicated because it involves three-way join and an additional two-way join. So we're going to retrieve the gene symbols from the gene product table of all the Drosophila melanogaster genes. I meant to have an additional constraint here. There are genes that are annotated to the nucleus. Okay. All right, so there's an example. If you look at the drop-down menu, they give you some examples of pre-written SQL statements to run these tasks. Okay. There was one that's sort of similar to what I'm asking, but not that I have additional constraints. But this would be a good starting point. Okay, so I'm gonna leave that open and show you what how we can change it. All right, so I didn't show you the inner join phrase, right? So when we join things, it's usually easier to say take one field from one table and then say equal to something else in another table. Inner join is a different way of writing the same thing. So I can take, let me take that inner join and rewrite it using our equal phrase in the where statement. So we will say from term, comma, graph path. So that's the table name. And then the condition that I have, I'm going to move it to the where clause as an additional condition. Okay. So that's the equivalence of inner join and the where clause. I can take all of these inner joins one by one and replace them with how I showed you before, or you can leave them as they are. It's just a different way of joining tables. Okay. All right. So this select statement contains more than the question is asking. The question is asking us to get just the gene symbols. So we're gonna have to get just the gene symbol and get rid of everything else. Okay, and let's call it gene symbol. So this keyword AS is a way to rename field names. All right, so you want to retrieve gene product dot symbol, but in the result you want the header field name to be gene symbol. All right. Okay, so we only get the gene symbol from this select statement. Let's look at the where clause. It links with additional tables whose information we are not really interested in, so we're going to get rid of them. We want to start with the species table, okay, and we also additionally want the species.species to be equal to melanogaster. Did I spell it right? I think so. Okay. So we want Drosophila melanogaster, and let me move this down. And we want the term.name, which is how the gene ontology database stores these annotations. They call them terms. Okay. We want the term nucleus, and the term comes from the association table. Okay. So we start with the term. We start with the term table, say term dot name equals nucleus, and then we use the ID of the of that term and go to the graph path table, match it with term1.id, graph path dot term1 ID. And now we're at the graph path table. We go from there. I'm just rewriting what's at the bottom. We take the term2 ID from that table and use it to jump to the association table. Okay. All right. And then from the association table, there is an association of that term with a gene product ID, meaning it links the nucleus to a gene name, and it'll have many gene names for the word nucleus. So we're gonna go from there, association table dot gene underscore product underscore ID equals, we're going to jump to the gene product ID, which lists the gene names, and we're going to link it to the ID field. Okay. So here's what we did. The second part, we start with the term name, which is the nucleus that we are looking for, and we search it in the term table. We find the row that contains the name nucleus, and then we take its ID of that row, and jump to the graph path table, linking with the term1 ID in that new table. We go from the graph path table using the term2 ID and jumping to the association table, linking with the term ID in that association table. Once we are at the association table, we get the gene product ID of those rows and link them with the ID field of the gene product table. And gene product is the table from which we are retrieving the symbol. So we are currently at our target table. Okay. But we need to let me delete these. So that's linking the term and a gene. But we have an additional constraint that we are only interested in the genes that are in Drosophila melanogaster. So we have a very similar thing. We start with the species table. We are interested in those rows that have the genus Drosophila and that have the species equals to melanogaster, which will actually result in a single row from the species table. And we want to go from the species table to the gene product table. And we do that by saying .id equals gene product .species ID. Okay. So we have two different join paths. One connecting the term with a gene, and the other one connecting species with a gene. And I will copy and paste this on the scratch pad, so you don't need to try to type, I guess. All right, so everyone cross your fingers, and let's see if it works. Couldn't run. Okay, so we have to inspect. I have an extra comma there. Let's get rid of that. Hopefully that was the only problem. I am missing an A and D here. Let's run the query again. Bad syntax. It usually would tell you what's bad about it. instead of just saying it's bad. All right. Okay, so what do we do? It says there's an error and we cannot find any obvious errors here. Can you see any obvious errors? Yes.
Do we have to do anything with gene symbol that adds, or is that just for
Yes.
displaying it?
So this one is, we don't need to do anything. There's a table called gene product. So we did some things in the where clause. We worked with some tables that we did not really declare that we're going to use them. So we have to list in the from all the tables that we are dealing with. So comma species. And we use the graph path, we use the association, we use the term, and we also use the gene product. Okay. And then let's say query. Yay. Okay. All right, so we get the results, but they repeat. Because, well, there may be some repeats in the database. And the way that sometimes you combine things causes some duplicates. A quick fix is to apply an aggregator function called unique, or I think distinct is the one that it might work. Let's try. I don't know if unique is a function in SQL, but let's give it a try. If it gives an error, we'll try the word distinct. And again, if you're not sure, you would search online for how to find unique entries in SQL. So the function name is distinct to take a set of values and return only unique ones, so that things are not repeated. Okay. All right. Now we get the same results, but without repeating something twice. Let's take this and where's the scratch pad? All right, so that problem is solved, hopefully correctly. Now let's move on to getting a database populated. All right, so let's download the file. Where is it? Where is it? Where is it? Let's go here. Let's go to the download page and then download predicted targets, save link as. Oh, I can actually open it here, let's see. It'll take a while to download. I should have something that's already downloaded, so let's work on that one. Okay, so it's a large file. So whenever you have a large file, it's a good idea to make a smaller version of it when you're writing your code, so that you don't have to run your code on the entire file and then realize you did something wrong, and then wait a couple hours to correct your mistake and rerun. All right, so let's get rid of everything here and keep just a few lines of microRNAs. You can also create fake data if you don't want to do what I'm doing. All right, so this is more manageable. I just want to have everything on one page. All right, and then let's save it as a different file so we don't have to, we don't write on top of what we have. Save as mirdb sample.txt. Okay, so we have a text file that has 3 columns, but it's not like an Excel file. The columns are they depend on how you interpret. This file is formatted using tabs to separate the fields. So on the first line, you have CFA-let7-let7a, and then you have a tab character. Okay, that's the character that comes in when you hit the tab key on your keyboard. And then you have the name of the gene, and then another tab character, and then a set of numbers dot a set of other numbers. So it's essentially a floating point number. Okay. So if you have enough memory in MATLAB, you may be able to load this in a single function call. All right. But if you can't, if there's not enough memory, then you would have to write a lower level function yourself, where you open the file, go through it line by line, and insert each line to the database. Okay. How many of you know how to go through the file line by line and parse the data? Okay. All right. So let's do that. Let me take this. mirdb sample.txt. You probably hate me for having that black screen, so let me just make it white. All right, so we have that text file. We want to write a computer program that will open the file and go through it line by line. So let's call that program. Let me go into the folder first. Edit mirdb underscore populate. Okay. All right, so to open a file, we will need to call the fopen function, give the name of the file, or its full path if it's not in the current directory. So we do that. That will give us a file identifier which we can use to refer to that file. And any operation that we do on that file will have to use that file ID. We want to open the file to read from it. The second argument would have to be W if you want to write into the file. Okay. Once we open the file, then we can do a series of fread calls. Okay. To read a chunk of characters. You can use fread to read the entire file into a string. There's another function called fget l to read a single line from the file. So let's highlight and then evaluate this in the command window. And then let's highlight this, evaluate it in the command window. Without the comma will show us the answer. So every time you do this, it's getting a new line, the next line from the file. Okay. So we will have to do this in a while loop because we don't know how many lines we have. And we're going to have to do this until we get to the end of the file. Okay. So we're going to have to say while we are not at the end of the file, and then get the next line. Let's save the next line in a variable called line. That's the variable that will contain a string. Okay. So at each point in this while loop, while line will be one of those lines. Okay, so let's do this in the command window. All right, so line is that. And this line has 2 tab characters. We can say line equal tab character, which you need to know the ASCII code. I guess you don't need to. You can just use sprintf to create the tab character. So let's figure out the ASCII code for tab. We don't see the result on the screen because it's white. It's number 9. Okay, but we don't need to memorize number 9. Instead of saying 9, we'll just say line equal sprintf \t. So that will give me a logical vector that has the same size as the line variable, and it will contain a 1 wherever there is a tab character. So I have the microRNA name in the first 13 characters, sorry, in the first 8, 9, 10 characters, and then the 11th character is a tab symbol. Okay. So this equal gives me the locations of the tab characters. It may be easier to take that logical vector and pass it on to the find function to get the locations of those tab characters. So on the line that I currently read, there's a tab symbol at the 11th character and at the 24th character. In MATLAB, you would use the, let me store that in a variable called tab locations, tab locks. I need to have 2 of them, otherwise my code won't work. Otherwise, that line is does not really contain 3 things. So I can take the line string, start from its first character, go all the way to the location of the first tab -1. So I don't want to include the tab itself. That's why I need to go all the way to the 10th character for this line. But for a different line, if the tabs are in a different place, the code will still work. Okay. So that extracts the microRNA name. So let's store that in a variable called microRNA. Okay, so let's do that. We got the locations of the tabs. We extracted the microRNA name. And then the gene name is going to be starting from the location 1 past the location of the first tab. all the way to 1 before the location of the second tab. Yeah. And that gives me the gene name or the refseq ID. Let's call that refseq. Okay. And the last thing is the score. That will be starting from the location of the second tab, plus 1, all the way to the last character of the line. That gives me the number that I'm looking for. Okay, but that's going to be a string. I need to convert it to a number. I don't want an array of characters. I want it to be a numerical value. There's a function called str2double that will take a string and convert it into a numerical double value. So let's store that in a variable called score. Okay. So as a result, from the line that I just read, I extracted microRNA name, refseq ID, and the score. And then this while loop, as it's looping, it'll do this for every single line. So now I need to push this to the database. Okay. Since I didn't really show you any way to connect to the database from MATLAB, let's just create the strings, and then you can just copy and paste them to in Firefox. Okay. So let's do sprintf to create a string. We'll say insert into, just as I should let you go. Insert into mir to target. That's the table name. And then let's specify the field names that we will store these values into. MicroRNA, comma, refseq ID, comma, score. And then values will have a string in quotes, another string in quotes, and a numerical value, let's say %f. So for each of those % symbols and their identifiers, I need to have an argument to this function. So %s will expect a string. The next %s expects me to provide a string. The next, the %f expects me to provide a number, score. Okay. And let's use fprintf so it just prints in the command window, and then you can copy and paste it from the command window. All right, so let's run this file by hitting F5 on Windows. You get the whole thing on one line. Let's put a new line character at the end of my format string in fprintf, and repeat this by hitting F5. And every time you run it, it runs the while loop, goes through the file line by line, creates that insert into statement, and shows it to you. And then you can take these, copy and paste them to the Firefox to run them. Okay? But do it only once, otherwise you'll get duplicate rows in your table. All right, so what I didn't do is I didn't really close the table. Whenever you open, I didn't really close the file. Whenever you open a file, make sure to close it. Okay, so this is how you insert in the table. Before you can insert, you have to create the table. All right. Let me create the table in here. All right, so we need to say create table, the table name, my mirdb. Okay, and then in parentheses I need to specify the field names. I'll have the microRNA name, which is going to be a string. Let's call it varchar, and let's be generous about how long it's going to be. 64 characters. Okay, it won't contain 64 characters, but let's not take any risks. You can figure out exactly what the maximum length is and then use that instead. You can specify up to 255 for a var card data type. So microRNA and then gene ref seq ID is going to be 255, and the score is going to be a float. Okay, so you just list the fields and their data types, and then when you run, you'll get that new table. Okay. I didn't want to. I already had the mirror to target. That's why I didn't name it that way. Any questions? This one should be named gene ref seq ID, and I will post this code also. All right, so we do have a way to connect MATLAB to databases, but it wasn't working for me earlier today. I will send it to you once I make sure it works. Otherwise, you can just create the strings in MATLAB or any other programming language and then paste them into Firefox. Or better yet, you probably should create a text file, write into that text file, and then load that in Firefox. Okay. Any questions? So my office hours are, I don't know when. I will send you the office hours. Okay.